Good morning. This is AP Biology review number 2. We'll be looking at proteins. This video will be a little bit shorter, and then we'll get into enzymes, a type of protein specifically, on video number 3. To start off with, if you're following along in your notes, this is in chapter 3. It's under Chem and Biochemistry. It's in chapter 3. We're down kind of on this part where it says 3.2 proteins and 3.2 amino acids. You might remember that proteins are kind of the doers of the cell. They're responsible for pretty much I always refer to them as the workers of the cell. Carbs are mainly storing energy or structure typically. Lipids, storing energy. Nucleic acids are primarily, even though RNA can have some catalytic properties, DNA and RNA are mainly for storing information. So at that point, everything has been storage. Nobody's done anything yet. And so proteins are the main doers, the workers of the cell. You might remember I always give the example in class of, pretend that we're at Timber Creek, who put up the walls at Timber Creek, who put in the windows, who tiled the floor, who put in the AC units. Those were all workers. People did those. And so proteins are generally the workers of the cell. So they run chemical reactions, enzymes. They can act in cell communication. They can be receptors in cell communication. So we'll look at those throughout the next few videos. For the moment though, I want to skip down and look at amino acids. So the genetic code codes for 20 amino acids. Now, there are some tweaks you can do to them to get a 21st and a 22nd, like selenocysteine and pyrrolysine and stuff like that, but there are 20 amino acids in the genetic code. And we group them. There's kind of about half of them, I think it's 11, or we'll just say about half, are essential versus non-essential. So essential amino acids you have to get from your diet. Non-essential, you don't have to. So if I ate nothing but sugar, that's pure carbohydrate, my body can make about half of the amino acids on its own, but about half it can't make on its own. So I'll die if all I eat is sugar. So that's why it's required that we get the additional amino acids in our diet, essential. If you ever hear of essential minerals or essential vitamins or something like that, essential means that you have to eat it. So the 20 amino acids, we generally group them into polar and nonpolar. I know that you can't see this picture down here right now. I've got significant stuff of it over here. But when we look at an amino acid, we generally group them into polar, nonpolar, big, small, kind of like that. So the general structure of an amino acid is this. I've got a carbon in the middle. This over here, COOH, is called a carboxyl group or a carboxylic acid. On its own, we call it a carboxylic acid. Once it's bound to something, we call it a carboxyl group. I mentioned this earlier in the video the other day. If we're looking at a periodic table, we're like, that's sodium. That over there on the right is chlorine. When we join them together, we call it sodium chloride. We don't call it sodium chlorine. So we change the name a little bit once it's bonded. So this is the carboxyl group or carboxylic acid group. And then this, NH2, is an amino group. You'll notice this carbon right here only has 3 bonds. That's because the remaining part is what we call the R group. It's variable. If we were in a math class, we would probably call it X, but we're not. So that's the general structure of an amino acid. Now, the structure of the amino acid changes based off of the pH of the environment that it's in. So you might see them where it says instead of C double bond O, OH, you might see one that has, instead of an OH right there, it might have an O minus because we donated our hydrogen ion, our proton. Over here, instead of having 2 hydrogens, you might have a third hydrogen, and then there'll be a plus, like NHH, then another H with a plus. Or you might find the halfway in between. So it all depends on the pH of the environment. So that's an amino acid. I might have gained a hydrogen, it's still an amino acid. I might have lost a hydrogen, it's still an amino acid. Okay, so the R group is what makes them different. That's an amino group. That's a carboxylic acid. That's why we call this whole structure an amino acid. It makes sense. You might think of amino. NH3 is ammonia. NH4 plus is ammonium. So whenever you hear those words, maybe from chemistry, some of those are kind of ringing a bell. Okay. In fact, whenever we break down amino acids, we end up producing ammonia. Ammonia is toxic to cells, and that's why we have to convert that into urea, urine, and then pee it out. Okay, so I've got this amino acid here. I've got different R groups that are possible. Again, I can break them down into 20 different categories. I know you can't see it on the screen. If you Googled, table amino acids, I'm sure you could find hundreds of tables of amino acids that bring up the 20 aminos. And they all have different R groups. So I might have that R group. That one is alanine. I might have this R group, or this R group, or this long chain, this R group right there. That's what makes each amino acid unique. About half of the amino acids are polar. Some are small, like that. Some are long, skinny. Some are big. Some are branched. Some are wide. And the reason that matters is because, just like last time, shape matters. So shape and chemical characteristic matters. You might remember from the lipids, whether it's saturated or unsaturated determines its shape, and that determines whether that lipid is solid or liquid, and whether it is more or less likely to clog my arteries. So shape matters. Okay, so if I wanted to join a couple of amino acids together in order to make like a chain of amino acids, a protein, I'd have another amino acid here nearby. I'll make it a different color so you can kind of tell what is what. Amino group. These groups are called functional groups just because we see them a lot. Because in organic chemistry we see C double bond O, OH a lot, we named it. That's a carboxyl group. This is an amino group like we said earlier. And then I've got some kind of R right here. Well, we also learned last time, anytime we want to join monomers together, small or single parts, monomer, mer means part, into a polymer, if I want to join many chains or many amino acids together, then I've got to do what kind of process? Yes, dehydration synthesis. So I've got to dehydrate, lose water, so that I can synthesize, put together. And that's exactly what will happen here. Those guys will join. That means I've got one bond too many. If this was stable, and this was stable, I know that this might bother you with octet rules and stuff like that. Don't sweat it. What we know is a moment ago, before we formed this bond, that carbon was stable and that nitrogen was stable. So if I add a bond to them, it just became unstable. So now I've got to get rid of one of these bonds. So if I get rid of this, now that OH is kind of on its own, get rid of this, now that H is on its own, then together they will combine to make HOH, which is water. So we just lost water, we just did dehydration, losing water, synthesis, built together, put these together. And then hydrolysis was how we split them apart. Hydrolysis is, I basically just take these arrows and go the opposite direction. I take a water, I lysis, split it apart, and then join there. Now this bond breaks, I just broke those down. Okay, so whenever I'm building a chain of amino acids, we kind of break this down into structures. You might remember that amino acids follow these patterns of structures. Like here we had some of these crazy looking ones like this. This was from our protein database online. All these different shapes. And so you might remember that there were levels of protein structure. So we had primary, secondary, tertiary, and quaternary. So over here, primary structure is what we were building just now. So if I join these together, and all of this is gone now, I'm building a chain of amino acids, hopefully in the right order. So primary structure is amino acids joined and then in the correct order. Sorry, my handwriting's not very good, but that says amino acids joined in the correct order. On past AP Bio tests, if you didn't mention the order, they didn't give you credit. So I know it's kind of picky, but what you gonna do? So I join them together. This bond is a covalent bond, but we also call it a peptide bond. So a peptide bond is just a special type of covalent bond. What makes it special It's joining together amino acids. Other than that, I don't know why it's special enough to get its own name, but it does. Okay, so I joined them in the correct order. And then secondary structure. We learned 2 secondary structures in class. Do you remember what they were? Alpha helix, beta pleated sheet. So once I have this chain of amino acids, let's say I have amino acid number 1 joined to 2, to number 3, to number 4, to number 5, to number 6, I can then start taking this string of amino acids, amino acid 1, 2, 3, 4, 5, 6, here you go. These are my amino acids, 1, 2, 3, 4, 5, 6, all the way to 100, 150 amino acids. And now I can start folding it, and it can start bonding with itself. So it might make kind of a coiled, almost like a slinky, or like half of DNA. So they call that an alpha helix. So when this structure starts to fold on itself and look like, so this is the primary structure. When it starts to fold on itself, like this, then we call that secondary structure. That one's called an alpha helix. If I look at this structure right there, I can see an alpha helix and an alpha helix and an alpha helix. This structure over here has a whole lot of alpha helices. Why do we name alpha helices? Just whenever you start looking at proteins, they're all over the place. So it's just a common shape that we see, so we named it. So that's secondary structure. I've got an alpha helix, and I've also got the beta pleated sheet. That says beta pleated sheet. And there, that's when my chain of amino acids is kind of making almost like a fan shape. Like if you ever took a piece of paper and let's say it was a hot day and you didn't have anything to fan yourself, you might take a piece of paper and just fold it back and forth like this, to where it makes kind of a fan shape like that, so you could fan yourself. Well, that's kind of what a beta pleated sheet looks like. Now what's interesting about this is up here in primary structure, amino acids are joined by covalent bonds, specifically peptide bonds. That's it. The R groups are not interacting with each other. That R group is not interacting with this R group. Once I start getting into secondary structure, then now I can have hydrogen bonding. If you look at your notes, we'll actually start breaking this down on the next page where it says 3D shape is everything. So we start having hydrogen bonding, but it's still only in the backbone. We aren't using the R groups yet. So what's holding that spiral together is hydrogen bonding between this strand and this strand, this strand and this strand. Those are held together, but it's still just hydrogen bonding at the backbone. So now we add in hydrogen bonding. So basically what we're doing is we're zooming out. It's kind of like we're on Google Maps and you're zoomed in on your house, then your neighborhood, then Tarrant County, then Texas or whatever. So we're kind of zooming out. We're super zoomed in. This amino acid is, phenylalanine and then histidine or whatever. Now we're kind of zooming out to look at how they interact. Then when we get to a little bit more zoomed out, now we're looking at how secondary structures start interacting with each other. So now I might have an alpha helix, and it might be chemically interacting with a couple of beta pleated sheets. Kind of like these ones on the screen. That alpha helix, that one, there's some beta pleated sheets that'll be difficult to see. Beta pleated sheets, whenever they draw them, they always look like arrows. And so when those interact, all bonds are possible. So we still have covalent bonds from the beginning. We still have hydrogen bonds because we got secondary structure. But we also have ionic bonds. We're now also including R groups. So now this R group and this R group might actually bond with each other. So between these, what's holding these shapes together? Some hydrogen bonds. Maybe there's a plus and a minus. That's ionic. Maybe I have some covalent bonds, so those will be solid lines in there. And that's kind of what's holding the structure together. So primary structure, fold it up into maybe an alpha helix. Maybe here's a beta pleated sheet. And then now those start bonding with each other, and more things start bonding with each other, until you get what at first looks like a tangled mess, but that might be a receptor and enzyme that has a perfect shape. Just like a lock and a key might look like a random cut on a piece of metal on the key, but it's not. It's a perfect fit for that lock that it's made for. Then, quaternary structure is whenever we have a new polypeptide joining into the mix. So we've got our original up here. I'll try to copy this. But then we also have a new separate strand. This is all one continuous line of yarn, so to speak. But now, if I have a new strand joined in, and they start bonding with each other, all bonds are fair game, pluses, minuses, ionic, hydrogen bonds, covalent bonds, then we call it quaternary structure. So now it's like primary, secondaries, those join to each other, tertiary structures, and then when I bring in a whole separate polypeptide string and they start joining with each other, then now we call that a quaternary structure. Just some random fun facts. Over here, when we start getting into R groups bonding with each other, when sulfurs and sulfurs bond with each other, those are really, really strong. And when they burn, they have a specific sulfur smell. So our burnt hair has a distinct burnt hair smell. Burnt rubber, if somebody peels out like their tires, has a specific smell that's kind of similar. That's from all the disulfide bridges. When an R group from here with a sulfur and an R group from here with a sulfur join with each other, they call it a disulfide, two sulfide, and they form a bridge. So bridge, disulfide bridges, add strength. That's why if you were to pluck a long hair, if you kind of pull it this way for as thin as it is, like if I had a hair, it'd be super thin, of course, but if I was going like this, like hairs are freaky strong for as thin as they are. And part of that is due to disulfide bridges. As far as anything else, let me double check to make sure I didn't forget anything that I wanted to mention before we mention Alzheimer's. Yeah, I think that's good. Alzheimer's. So Alzheimer's, not old timers. I don't know why some people say that. Alzheimer's is actually named after the doctor that discovered it. Or was it the name of his first patient? I'm not sure. It was either the doctor or the first patient. So with Alzheimer's, brain tissue, here's kind of a cutaway, like right here, if we just cut my face off and remove it and you're looking at my brain, normal healthy brain, advanced Alzheimer's, you'll notice it's smaller. Here's an actual medical image. This is if I turned and looked this way. So with Alzheimer's, what typically happens is you start losing brain tissue at the frontal and prefrontal lobes, the front. And the front is the main part, from here forward is the main thing that makes you different from a rat. From here back, you're just a giant rat. From here back, you're just controlling your blood sugar, digestion, respiratory rate, all the standard things that every other mammal is controlling. But what makes us different is this front part is where kind of the human intelligence, we'll say, comes in. And so you'll notice that right here, if I just find, they call them a gyrus and a sulcus, don't worry about it, we'll just call them hills and valleys. Here, the hills, the mountains are really wide, and the valleys not so much. Here, the valleys are getting bigger and bigger, and the hills are getting more and more narrow. It's not supposed to look like that. One way to think of it is if I covered this up, and you're trying to picture what the rest of the brain looked like, you would not have thought that, right? Big spaghetti, small spaghetti. Sorry for the food analogies. Here's another one. This one is even a little bit more obvious. If I were to say, what does the rest of this brain look like? You wouldn't have thought that. Look at how wide those mountains are versus how wide those mountains are. So Alzheimer's. Well, we don't know for sure what causes Alzheimer's. I'm going to tell you what we do know. We do know that it's probably linked to human chromosome 21 because for us, I don't have Down syndrome. So about 3% of 65 to 74-year-olds have Alzheimer's, but about half of those, 85 and older, have the disease. The average age of diagnosis is at 80. However, Here's a link from the Cleveland Clinic, and it says that if you have Down syndrome, about half of people with Down syndrome get it in their 50s. So 3% in our late 60s, early 70s, or half of us in our 50s. So we think that it's linked to chromosome 21. Now, what's the cause of it? We know and we don't know. So here's a normal neuron, or set of neurons. There's 1, 2, 3 neurons. I'm going to double check real fast just to see what you guys can see. That'll work. And so actually let me zoom that in just a little bit. Get my almonds out of the way. These are my props. So 1, 2, 3 normal neurons. With Alzheimer's, we see these neurofibrillary tangles. Neuro, nerve cell, fibrillary, fiber, tangles. Those are inside the nerve cells, and we also see these protein plaques, amyloid plaques, outside the cells. So here's what it looks like under a microscope. These are the neurofibrillary tangles. Neurofibrillary tangles are made out of this protein called tau. So that's a nerve cell, and that little thing that looks like long hair, those are neurofibrillary tangles like squirting out of the cell. You're looking, let me keep clarifying this, you're looking down the barrel of a nerve cell. So let's say my neuron, my nerve cell is, a long tube like this, well now you're like looking down the barrel of that tube. So here, what we'll see next, I've got microtubules, and they're supposed to be stabilized by tau proteins. That's normal. But in patients with Alzheimer's, we start to see that those tau proteins just sort of tangle together, and so microtubules aren't stabilized. And now there's this kind of mess of tau proteins. And then again, under the microscope, it'll end up looking like that. Here, we can see the amyloid plaques. So amyloid plaques are something separate. All these little things right here that look like, I don't know, they look like amyloid plaques. These are proteins that build up outside the cells. Again, we don't know, is Alzheimer's causing the tangles and the plaques, or the plaques and tangles causing Alzheimer's? We tend to think the plaques and tangles are causing the Alzheimer's, but sometimes it's hard to tell what's the cause and what's an effect. We just know that they're currently correlated. So the reason that I bring that up is that shape matters. So whenever I have these chains of amino acids, I'll have DNA that codes for mRNA, and then that mRNA codes for the order of amino acids. If they're in the right order, the right primary structure, then they should fold up into the right shape and do the right thing. But if they don't, then they might end up making a mess. So my DNA, pretend that the string behind me doesn't exist yet. My DNA is coding for RNA, which eventually codes for amino acid number 1, 2, 3, 4, 5, 6. Let's say those first 5 amino acids are negative, and the next 5 over here, let's say I have 5 here that are negative, a few that are neutral, nonpolar, and like 5 that are positive. Well, as this is being built, it's just kind of flailing around randomly. Negative won't stick to negative, negative won't stick to nonpolar, but negatives and positives will stick. So as I'm building this protein, it's folding into the right shape. I also have other proteins, ironically, that help fold proteins. Okay, so eventually I have this folded into the right thing. What if my DNA, though, has an error? Well, if I'm supposed to have 5 negatives followed by 5 positives, let's say that I substitute one of these positives for a medium-sized, medium positive one. I might still fold up into the right shape and be okay. We call that a silent mutation. But what if I've got these 5 negatives, and over here I put in a few negatives? Now they won't stick. So now my protein is going to be misshapen. Now I might have a mutation that actually has some sort of consequence. So DNA codes for the order of amino acids. If the order is correct, we've at least got a shot at folding into the right shape. But because of the chemical and physical characteristics of each amino acid, if I'm supposed to have negatives here and positives here, great, they stick and they form a loop. That looks like the beginning of a beta-pleated sheet. But if that was negative, that was negative, that was negative, that's a negative, then nobody's going to attract to each other, and now I have a mutation. And it could be catastrophic, or it could just mean that now I don't have brown eyes, now I have blue eyes. So the last thing that we want to look at in our notes is denaturation. So there's denaturing a protein, and then a protein can also do a conformational change. Those are different things. So whenever we denature a protein, let's say I have this protein that is going to make some kind of enzyme or some receptor that looks like that. So that's my primary structure, secondary, tertiary. I don't see a second strand, so I don't have quaternary structure. Okay, well, it might be that this particular enzyme, so I'll put E for enzyme, is supposed to fit this particular substrate. All right, great. Well, whenever it's going to fit, I need, let me redraw this. It's not going to be identical, obviously. Well, whenever I'm doing that, if I denature my protein, then what kind of things? We had two major things that can denature a protein. That's right. Temperature and then ion concentration. And I'm lumping ion concentration and pH together. So if you want to say pH is a third one, then you can say 3. So the way that heat denatures proteins is what's holding these secondary structures together. Hydrogen bonds. Covalent bonds are really strong. It's hard to break those. But hydrogen bonds are fairly weak. So that shape and that shape, my alpha helix and my beta pleated sheet, are being held together with hydrogen bonds. So whenever this protein that's supposed to work at body temperature, let's say 98, 99 degrees, at 98 or 99 degrees, this alpha helix is kind of vibrating. That's what things do at temperature. At 100, 101, 102, 103, 104, they vibrate more and more and more. Somewhere around 108, 110, 112, proteins, essential proteins in your brain cells will actually vibrate so much that the hydrogen bonds break. And when that happens, your protein is no longer its natural shape. It's not natural, it's denatured. And if we cool you back down, then now those hydrogen bonds might go back together, but they might not go back together in the right spot. Keep in mind, when I was first building my protein, and this part may be hydrogen bonded with this part, and then this part hydrogen bonded with this part, and this part hydrogen bonded with this part, everything's cool. But now, once I've built the whole protein, if it denatures and unravels, and then I cool it off, when it goes back, who's to say that this part doesn't hydrogen bond with this part over here, or this part over here? So the likelihood that it goes back to the correct shape is typically unlikely. So as a general rule, when you denature a protein, it's ruined. Now, we'll call, whenever we break the hydrogen bonds in DNA, we call that denaturing, and then they can go back together, like in polymerase chain reaction. So it's not always ruined, but generally when we think of proteins denaturing, they're ruined. And the example that I've always given in class is if I had an egg, pretend that this is an egg, I crack it and I just drop it on the counter, it'll be like clear and runny with like yellowy yolk in it. But if instead I drop it into a hot frying pan, the clear runny stuff turns into white and it turns solid. That's because I denatured the protein in egg. Then if I cool the egg back down, it doesn't go back the way it was. It doesn't go back to clear and runny because I've changed it forever. So it denatured, formed new bonds that are stable in this new environment, and now when I try to take it back, it doesn't go back to the original form. That's different from a conformational change. A conformational change is a reversible protein. So when we think of like proteins that might be like gates on sodium ion channels, for example, in the nervous system, they have formations where they can kind of open a gate or close a gate. If I have a cell, and over here I've got a gated channel, a conformational change can open and close that door. So I can open it or close it. I didn't ruin it when I opened it. I didn't ruin it when I closed it. A conformational change is a reversible change. So heat can denature proteins. Whenever I freeze proteins, the reason that's, and by the way, That's why we die if we get a fever that's too high. We denature vital brain proteins and now we're screwed. The reason that I freeze to death is not because I'm denaturing proteins. We know that freezing doesn't necessarily destroy the proteins always, because I can take frozen peas, throw them in put them in like some soil with some water, and they can grow. Whenever they ship enzymes to us, they stay cold. We freeze them until we're ready to use them in lab. So the reason that freezing can end up in death is not denaturing. Freezing is because the enzyme and the substrate, for example, aren't colliding enough. So at 99 body temperature, substrates and enzymes are bumping into each other, let's say 500 times a minute, like this. But at 90 degrees, they're only bumping into each other 480 times per minute. And that's not enough to keep us alive. So that's why you freeze to death. Another way that we can denature enzymes is if we have a whole lot of like H plus floating around. That would be in an acid. Acids begin with H, or at least that's what you learn in chemistry. So HCl is hydrochloric acid. H2SO4 is sulfuric acid. H3PO4, anything that I see with an H at the beginning, based off of your chemistry class, you're like, that's probably an acid. Generally, if something ends with an OH, sodium hydroxide, potassium hydroxide, ending in OH means it's a base. Once we get into organic chemistry, that's not true. This is a carboxylic acid, and it's got an OH. So a better definition is anybody that gives up hydrogens, proton donors, hydrogen ions are protons. Proton donors are acids, proton acceptors are bases. So here's what this means. If I take this protein and I put it in hydrochloric acid, if I dumped hydrochloric acid or battery acid into my eye because I'm an idiot, then the reason that that's going to denature those proteins is all of a sudden I've got a bunch of loose H pluses. What's holding this structure together? Again, I've got polar interactions, a slight positive and a slight negative, hydrogen bond, or a full positive and a full negative, ionic bond. Well, if all of a sudden I've got a bunch of loose pluses or a bunch of loose OH minuses, hydroxyl groups floating around, then they can start attacking those bonds. And so now, this and this won't be attracted. Now, this guy will bond to the H+, which means I just changed the shape of my protein. And so that's how like an acid burn can denature proteins. That's why if I get acid in my eyes, I'm denaturing proteins. And if if I'm not lucky, then I'm gonna lose vision, I'm gonna go blind. Let me see if there was anything else on denature. The last thing, since the whole reason we're meeting like this is because of COVID, this is the idea behind bacterial or viral. By the way, COVID is a virus. The 2019 version, COVID-19. And this is how like aseptic or sterilization, things like that work. If I've got bacteria on a piece of steak and I eat it and it's raw steak or something, then I might get sick from, E. coli or whatever the bacteria is. But if I throw that steak on the grill and I raise the temperature enough to denature the proteins, I might even actually toast the E. coli. Now I kill those E. coli. So grilling and cooking doesn't remove the bacteria, it just kills them. Same thing with viruses. They aren't alive, but soap and water can wash them off of your hands and destroy them. Alcohol at a high enough concentration can destroy them. And so that's why if you have something like viruses on your hands, you basically are trying to find, okay, how can I make an environment to where they won't survive? And the reason it doesn't destroy you is your outer skin cells are already dead anyway. They're just full of a protein called keratin. I know keratin's a protein because it ends in IN. Protein's usually in an INE or ACE. And so basically I just have a bunch of keratin in my cells to make them waterproof. So this whole outer layer is dead. So when I start putting soap and water on it, it doesn't really matter. I'm not killing my own cells because they're already dead. I think that is it for today. Again, our next session, we'll actually dive into how protein structure relates to enzymes, exothermic, endothermic reactions, feedback, how do we inhibit enzymes if we want to turn them off, stuff like that. Thank you very much.